Our next speaker today is going to be Ron Hardy. Ron is an expert in production fish aquaculture and has worked in the nutrition of production fish for years. He has an incredible expertise in this area. Came from University of Idaho, and you're still pretty active there these days, except for the boat trips to the San Juan Islands, I understand.
Which I should be there right now.
You should be there right now. One of the things we wanted to bring into this session was that fact that production fisheries or production aquaculture has been studying nutrition a long time. It's paramount to the success of those industries that we understand the nutritional needs of fish that you're going to try to eventually put on your plate and eat. That being the case, that expertise can be transferred even into small fish, which we generally won't eat as zebrafish, but a lot of that information can be transferred, and Ron agreed to come and tell us a little bit about the history of some of the production fish and perhaps give us some insight into moving forward. Ron, thank you.
Just a reminder, these little black pop-up things are your microphones. So try not to cover them up.
Thank you, Steve. The topic that I was given, as Steve described, and thank you for inviting me, by the way, was a role of nutrition research in developing production aquaculture and how this might be applied to zebrafish, as he said. All right, Ron, let's see here. I'm off to a good start here, aren't I? All right.
The other way.
Okay, now I'm in business. So I thought I'd begin by just giving you a slide here showing you the growth of aquaculture over the last few decades. And the reason I'm showing you this is to illustrate how rapidly it's grown, but also when you look at these numbers, I'll explain in a second, realize that this growth has been supported by development of diets and a very great increase.
Excuse me, you get to the microphone.
Sorry about that. Can't see my slides very well over here. It's been accompanied by a tremendous increase in the amount of feed produced by the aquaculture industry. So just to explain this slide a little bit, what you see in the increasing numbers there is the amount of fish that are captured in the wild, which is illustrated by the white and the green areas, and then the growth of aquaculture over the last 20 or 30 years in the red. And you have to discount the part at the bottom, the green area there. It refers to industrial fishing, in other words, fish that are caught and not used directly for human consumption. And then the purple line shows the per capita fish supply worldwide. So you can see it's been on an upward trend. And you can also see that over the last, since about 1990, aquaculture's proportion of the amount of seafood or fisheries products that are consumed or produced for human consumption has really grown a lot, whereas the amount that's harvested from the sea has been pretty flat. So we pretty much tapped out all of the resources that we harvest from the ocean. Another way of showing the same data, but it's a little bit different slant here. Aquaculture, we like to go around and tell everybody, it hasn't really resulted in any big money being sent our way, but anyway, it's been the most rapid growing livestock production sector in the world for the last 20 or 30 years. Since 2000, FAO says it's grown 5.8% per year. It grew a little faster before that. And as I said, in the yellow there, you can see capture fisheries has pretty much been level since the 1990s, and aquaculture has increased tremendously, just taken off like gangbusters. Latest data shows about 76.6 million metric tons of fish. Now, aquaculture includes seafood, shellfish, and other things, but that's just fish. So it's really gone crazy. Why has this happened? Well, there are a number of reasons. I'd like to say it's all nutrition, but it isn't. Vaccines have been a great development in that they've eliminated or at least reduced the losses of fish to viral and bacterial diseases. We've seen a lot of genetic improvement in some species, particularly salmon and tilapia, selecting for growth and for other performance. Government policies, this really refers here to China. If you look at the little circle in the bottom, you can see China accounts for almost 2/3 of all of the fish production in the world. And if you can see North America there, we're 1.2%. We're not very big. But, and this is my big but here, better feeds and nutritional requirements. That's played a huge role in the development of aquaculture and expansion. So anyway, early on, I'm going to give you a little history here. Fish nutrition research was really based upon not a lot of fish, but mainly the need to produce salmon and trout for what we call enhancement hatcheries. So these are hatcheries that grow little baby fish, release them into the wild, and they go to the ocean to complete the life cycle, come back, enter the fisheries, and are captured and contribute to economic activity. Only in the last 30 years, when farming really took off, has the need for really well-formulated feeds taken place. And what, if you think about it, step back, fish food production, this growth of aquaculture has really changed the type of research that we do. When I started in this business, all we cared about was producing little baby fish, smolts, and trout for enhancement and release. But now, with food production, not only has the thrust changed, but also the economics of production has changed too. The reasons, the goals of production. The goals of production used to be, let's produce a really healthy fish that's going to survive after it's released. We don't care what it costs. We only do this once a year. The big goal is let's really not lose any fish and make sure they're healthy. Now, let's make money. That's really what the name of the game is. So a little historical background. We're all researchers. We all chase grants. In 1924, these two gentlemen, Embody and Gordon, did, I think, one of the most fun research projects, at least it sounds like fun, that was ever done. They were sent on a fishing expedition. They went from upstate New York all the way to Minnesota fishing all summer. And what they did was caught trout, caught other fish, collected their stomach contents, took them back to the lab, analyzed them to see what is it that trout consume in nature. And after they got done with all of their analyses, it averaged out to be about 49% protein. And I noted here there was no adjustment for chitin because, trout eat a lot of insects. So that's maybe a little high. Fat 15 to 16%, fiber 8%, ash 10%. That was considered then the goal or the template or what you would try to achieve in making a feed. Oh, geez. I thought I got all these stupid things out of here. Okay, so 1920s to 1940s. Pretty much in those days, there weren't a lot of commercial companies. Salmon and trout feeds were made at the hatchery by the staff. They sourced locally available materials. They made these wet and dry mixes that were kind of pretty primitive. They were formed into clumps or noodles. There was really no sophisticated pelleting taking place. And they were made from whatever these people could scrounge around. They went to slaughterhouses and got animal organs and animal parts, old horses, honest to God. I met a guy who used to grind up horses. Carp, suckers, other fish that nobody ate, dry blends of other byproducts, dried milk, yeast, and wheat. Those were the kinds of ingredients that were used. All right, Ron. Oops. Okay, Ron, let's go back one. So, to summarize this, a lot of development of these diets led to this type of formulation. Whitefish meal, 40%, cottonseed meal, wheat midge, brewer's yeast, dried skim milk, fish oil. This is, I've simplified this just for purpose of this talk. Having a crude protein content of 49%, this would be a dry diet, fat, ash. So you see it kind of reflects more or less what our boys from Portland had done in 1924. And then, anyway, prior to this, or prior to the 1950s, all of these feeds that were developed were really done by trial and error. They were empirically derived. We do a little bit of the qualitative dietary requirements of some nutrients, but not much about the quantitative requirements. And what really was needed at that time was what you've been talking about here, or what Frosty just discussed, and that is the need for a purified or semi-purified diet could be used in research. There have been a lot of attempts to do this, but none of them had been successful for more than a short period. They were all nutritionally inadequate. But then again, what happens? The right person came along at the right time and moved the field forward, and that was my professor and the guy considered the father of fish nutrition, John Halver. And I call him the accidental fish nutritionist He had started out to become a human nutritionist. He was a student at the University of Washington in a PhD program. And at that time, in the early 50s, there was a fisheries guy named Donaldson, who was just a giant in the field. He came to the chairman of the Department of Biochemistry. He said, I've got a problem with our fish. Our fish are anemic. We don't know what the deal is. We don't know how to cure it. It's really a problem. And the chairman of the department yelled out the door, Halbert, get in here. This is what he told me. He said, this guy needs help. See what you can do. Now, just at that time, folic acid and B12 had been recently identified and purified, and they happened to have some. So Halbert went down to the little hatchery, injected these fish with folic acid and B12, and lo and behold, in 7 to 10 days, the anemia was resolved. Halbert's pretty smart. He thought, wow, these guys don't know anything. This is an opportunity for me. Maybe I won't be a human nutritionist. He proposed a PhD project to develop a vitamin-free test diet to study vitamins. He did this based on casein, gelatin, dextrin. It worked. He then used this diet to determine the qualitative and then quantitative requirements of Donaldson's salmon. I got a hold of his original mix sheet. I know you can't read that, but it looks like a crazy recipe out of a cookbook from the 40s or 50s. Mix this, one part of that, put on a yellow hat, do this, do that. But boiled down, this is what it looked like. It had vitamin-free casein, dextrin, and gelatin, wheat starch, CMC, alpha cell, vitamin minerals, amino acid mixture, choline chloride. This was a moist diet. It was made into noodles. It was kept frozen, and its crude protein was about 34% on as-is basis, fat 17, ash. Wait a minute, maybe that's not right. Well, anyway, that's what it looked like. So what Halbert did, the semi-purified diet was, you know how this works. Make this deficient diet, make your vitamin premix, leave out the nutrients you want to study, add graded levels, in Halbert's case, 6 to 8 levels. Feed the fish till they gained about 3 times their initial weight, if possible. That didn't always happen. And then measure response variables. Now, we're talking the 1950s here. So their response variables were relatively simple: weight gain, survival, feed conversion ratio. A little later, they developed microbial assays with which they could analyze for vitamin levels in livers. A little later, they got active and developed some enzyme assays, histology. But remember, at this time, what was it? No HPLCs, no GCs, no mass specs. This was all done at a pretty simple level. So this is the kind of stuff you would get. This happens to be a microbiological assay of thiamine. And you can see the statistical analysis was a broken line approach. And this is the kind of data that was generated during that time, showing, in this case, a maximum liver thiamine content at about 8 milligrams per kilogram diet. This diet was modified to then work to study amino acid requirements. In this case, vitamin-free casein, gelatin were reduced, and amino acid mixture was added, and away they went, leaving, of course, 1 amino acid out at a time, same diet, basically. And this is the kind of data they got. So this case, this is the arginine requirement. You can see the fish started about 2 grams in average weight. The dark line is the deficient diet. They lost weight slightly over the course of the 10-week experiment. The fish that had arginine grew at a fairly nice rate. They didn't quite make the goal of 3 times their initial weight, but they did grow. And at 6 weeks, Halbert split his deficient group into 2, replacing arginine in the one group of the deficient diet. And you can see here, they started to grow. So that's the kind of study that was done back then. So using this approach over years of study, the nutrient requirements were more or less worked out. 10 essential amino acids were identified. Omega-3 fatty acids were shown to be essential. Energy mainly supplied by lipids and protein. This is in salmon. Carbohydrates really weren't that good as a carbohydrate, as an energy source. 15 essential vitamins, minerals, carotenoids subsequently were shown to be needed for viable eggs. And of course, minerals. There are other minerals that are essential, but because, as Frosty mentioned, fish live in water. A lot of these minerals can be obtained directly from water. So this is the NRC requirement for salmon fingerlings, I might add, showing that they're pretty well worked out. Nothing too exciting here, but anyway, there they are. That's the kind of formulate, or that's the data that now, still this to this day, is used to formulate commercial feeds. I want to give a credit here to work done at Oregon State University by a couple of guys I knew, John Costell and Russ Sinhuber. They did very, very interesting work on omega-3 fatty acids, showing that they needed 1% of the diet as omega-3s. These can be EPA or DHA. They also identified essential fatty acid deficiency signs, including heart pathology, fainting. Have you ever seen a fish faint? Probably not. Anyway, poor growth. It took a long time to make these fish deficient. Previous diet history had quite a great effect on the onset of deficiency, how long it took to get it. And then later on, this requirement was confirmed with foods, confirmed with Pacific salmon. So Haber assembled all this material and published his first book in 1972. This isn't it. I loaned mine to a guy who took it to Vietnam, and it was eaten by ants. I swear to God. So I lost my first copy. Anyway, this is the third copy. Howard and I did this together in 2002. But his first book was really a revelation. It was the first real treatise on fish nutrition, and it really broke from tradition in the fish nutrition world in that it wasn't written by a bunch of fish biologists. It was written by a biochemist. So it really had a biochemical approach. It established fish nutrition as a legitimate academic topic, and we are just starting to work on a fourth edition of this book. In 73, a year later, out came the first NRC bulletin on fish. That's the green one here, and subsequent bulletins have been issued in up to 1993 was the most recent one, up until the most recent one, which I'll talk about in a minute. That being this book right here. And two of the people that worked on this are in the room. Del Gatlin and Lou DeBramo were on our committee. So thank you for that. Thank you for your service, as they say, on behalf of Fish Nutrition. This was published in 2011 and is the most recent compilation of nutrient requirements of fish and shrimp, although it's getting to be a little old already. Now, here's what I see as a problem you have with zebrafish. Here's a copy of one of the tables from this book showing the freshwater fish requirements for amino acids, and you can see I compared in this slide the old 1993 and the 2001 or 2011 version, different colors there. Channel catfish, we know a lot about the amino acid requirements of catfish. We know a lot about rainbow trout. We're getting to know a lot more about common carp and tilapia. These are both very important commercial species. Zebrafish, nothing. We got nothing. Vitamins, same story. We know pretty well. You can see if you really want to dig into this in detail. The requirement levels did not change too much in our new bulletin. They're pretty much the same. A few little changes, little tweaks here and there. Again, zebrafish, not much there. All right, you've seen this already. This is minerals. Do I have to explain this one? No, we get the picture here. We don't know too much. Okay, one of the outcomes of the development of these data for nutrient requirements and application of this information in the production of commercial diets has been a profound change in the approximate composition of feeds for our salmon and trout, or this is trout in this case. And you can see since the 70s, the level of total protein in blue has increased from mid-30s up to the low 40s. But more importantly, the level of digestible protein has increased, and the gap between total and digestible protein has gotten smaller. The other thing to note is that the level of dietary fat has gone up a lot. And currently it says here 22, this slide's a little out of date. People are even pushing 30% now in some diets. Oh, dang it. And what's happened in commercial production is that this has really changed the economics of production. 1970s, the food conversion ratio of salmon and trout was over 2 or even higher than 2. And over time, with the improvements of diets, you can see it's progressively decreased such that we can now see, well, this is 2000. Boy, I got to update these slides. It's now down around 1.2 for trout and about 1 for salmon. 1.2 for trout and as low as 1 for production of up to 3 kilos salmon. So it's a tremendous improvement from where we were before this information was used to formulate feeds. So now I'm going to switch gears a little bit. How many of you took sociology when you were in college? Everybody did. Everybody knows what this is, Maslow's hierarchy of needs. So you know that probably from your reading that the hierarchy of needs starts out very basic. The basic requirements for survival, physiological needs, food, water, air, shelter are the bottom level. Safety and security needs, social needs, friends, family, community, recognition. And eventually, you really are lucky and good, and a lot of effort, you can become self-actualized. Well, I've created something called the Hardy hierarchy of feeds. So, I just dreamed this up here to kind of show some of the components, some of the elements that relate to our eventual goal of producing sustainable feeds in commercial production. So again, we have similar setup here. Nutritional requirements to sustain life are at the bottom. As we get more sophisticated, we can refine diets to supply nutrients needed for optimal growth and fish health. Feed ingredient knowledge is a component of this. It leads to more sophisticated feed formulations and eventually to sustainable feeds. So here's how I see it. I think trout are up there we're doing pretty well with trout. We're up there where we can formulate very sophisticated feeds, and we're almost to the point where we can produce sustainable feeds. I'm sorry to tell you this, your zebrafish are way down there. You have diets that seem to me capable of sustaining life, but as far as the nutritional needs for optimal growth and health, I don't think you've gotten there yet. So with trout, we still need to learn some more information, particularly on the nutritional needs for optimum growth and health using gene expression and other immunological parameters. We're getting a better idea of the effect of diet and nutrition on health. Feed ingredient knowledge, we have developed extensive databases on digestibility of nutrients in a whole array of common feed ingredients that really help us when we get to the next level, which is feed formulation and understanding better interactions among nutrients and ingredients. And like I said earlier, we're almost production of sustainable feeds is really within our reach. We can see it coming in the next 5 or 10 years. Oh boy. I had maybe one too many coffees this morning. So with zebrafish, here's what I see. Nutritional needs for optimal growth and health, not too much is known. Feed ingredient knowledge, some is known. You know some ingredients are good and bad. But when you get to feed formulation and feed ingredient interactions, it's almost impossible, if not difficult anyway, without knowledge of nutritional requirements. You just can't do it. Meaning you cannot achieve optimized diets for whatever purpose, medical research, physiological or developmental studies, and so on. So why is this important? I think you all realize this, and Frosty alluded to this. It's really impossible to formulate feeds, nutritionally balanced feeds for zebrafish if we don't know what the nutritional requirements are. You just can't do it. And we all know that feed ingredients affect physiological functions. Again, this was said earlier. There are just a couple examples that we know there are signaling nutrients that affect energy allocation, protein accretion, and so on. We know that some of these ingredients contain phytoestrogens, fine ingredients in particular. Insect hormones are kind of an unknown, but may affect reproduction. And as Frosty said, Frosty really said a lot of things that I was going to say, so I guess that's good. We'll repeat it again. And that is, feed ingredients vary in nutrient contents. They vary with season, they vary with source, they vary with raw material quality and processing methods. The most variable ones are fish meal, insect meals, and land animal meals. And fish meal, I tell you, it's an amazing product. It's a very complex material. It contains all of the essential nutrients, or not all of them, but many of them that we need. High energy content, but it also contains other biologically active compounds, anabolic steroids, organic pollutants, heavy metals. Insect meals are interesting. Insects are like balloons. If you feed them, they basically take up whatever you feed them. So if you feed them certain fatty acids, they're going to accumulate those fatty acids. So they're almost like a blank canvas, like a balloon you fill up full of nutrients. So it's important to know what substrates are used to raise these insects, and of course, what species you're talking about. So insect meal is not a commodity like casein, or at least a defined ingredient. It can be a lot of different things in terms of nutrient content. So zebrafish diets, we know that there are closed formula diets that are commercially available from feed manufacturers. Some are designed specifically for zebrafish. Others are designed for their species, but work okay with zebrafish. They are kind of at the bottom level of our hierarchy of needs. They support decent growth and health. They don't kill a fish off right off the bat, but they're not specifically designed for zebrafish. And then in my perusing the zebrafish literature, I see there are practical diets that are used that contain unrefined ingredients, such as the ingredients I mentioned: fish meal, insect meal, grains, oilseed meals, not to mention the starches. And then we also have semi-purified diets that contain highly refined ingredients: casein, gelatin, wheat gluten. I found this in the literature. They got some nice diets there. But as a fish nutritionist coming from the commercial sector, I was kind of surprised when I looked through the literature in preparing for this talk at some of the stuff that caught my eye. And again, Frosty said this, he's absolutely right. Insufficient information on experimental diets is a major surprise to me anyway, in looking at some of the published papers in the zebrafish literature. And what do we need there? We need to know feed formulations and sources of ingredients. We need to know chemical analysis, not every chemical on the face of the earth, but certainly the ones that are important nutritionally. And just to let you know, as an editor of a scientific journal, and I think Delbert is an editor, I know Delbert's an editor, and he probably has the same opinion as I do, this information is required by aquaculture journals. If I get a paper submitted to me that doesn't contain this information, it's rejected at the editorial level. Goodbye. We don't even consider it. The other thing that surprised me was the source of commercial feeds is often not given, other than, oh, we bought a flake diet, or we fed worms, or we did this, or we did that. That surprised me too. The other thing that surprised me was that many of the papers that are published in this field are published in the journal Zebrafish. Guess what? That's not accessible to people like me, unless I want to shell out $51 to get a reprint. I was surprised that isn't open access, or at least accessible by normal or commonly used literature search engines. So there you go. So in the interest of being politically correct, I took this guy's name off this paper, but I thought I'd give an example of what not to do. This is a paper published in 2017, pretty recent paper. Point was to study carnitine. It was done in China. I know you probably can't read all this, but what the experimenter did was he bought a commercial zebrafish diet made in China. He gave the source of who produced it, some company. That was it. Not another word about that diet. He then took dough flour, dough he called it. I dug around and found out that's wheat flour. Mixed up carnitine by hand, dried it out, and then his experiment, which is the lower level of these little boxes here, where he would control feed, which was 1% dough and 3% of his mystery diet. And then a control fasting, he just fed the dough, and then he fed the dough with carnitine and the dough. Anyway, mix and match. Interesting study, but how do you know what's missing here? The diets weren't analyzed for carnitine. What's in the Chinese commercial diet for carnitine? I don't know. Didn't say. Didn't analyze it. So that's something that we would probably ding right off the bat if it was sent to my journal. I got a good example here from Oregon State University. Let's hear it for Oregon State. I read this paper. I gave his name because he did a good job. So this was an example. If you want to see the kind of information that should be in these papers, this is the one to look at. The authors presented complete information on the experimental diet, the whole formulation, the percentages of each ingredient, the sources of each ingredient were given. The mineral and vitamin premix were completely described. Chemical analysis conducted, results were presented, and he was looking in this case at putting tocopherol and analyzed all the diets to show that the added levels were pretty close to what was analyzed and found to be present. So that's what you want to do. Well, some of the uncertainties, I mean, these are common. You can figure this out. You know this. That's why we're here. We don't know the nutritional requirements of these fish. They likely change with life history. Somebody mentioned strain or genetic variants. We don't know that. We don't know deficiencies or antagonistic interactions that confound results. We commonly run into this stuff in commercial diets and into this problem. And it's particularly important when we're looking at gene expression studies. We can find minor changes in diet composition can alter gene expression and activation of certain pathways. Feed ingredients contain compounds that can affect metabolism. Obviously, reproductive effects. We've seen this in some. I've actually did a zebrafish study one time and changed their reproductive status. Redox status was mentioned earlier. And then another thing that was mentioned earlier, this guy mentioned a lot of good things here. I'm just going to give him credit here. Frosty did. And that is diets can vary in nutritional content from batch to batch. Close formulations may change. Producers can make small changes in using different substituting ingredients or altering the level of the same ingredient. Small changes. And more importantly, people don't realize this. A lot of these ingredients that are used, not the highly refined ingredients like casein, but the natural ingredients, shall we say, can vary a lot, and this may affect your results. So going forward, best practices should be followed. Just to repeat what I said, zebrafish studies should include a commercial reference diet to facilitate comparison among labs or between labs. Excuse me. I think studies should conclude an open formula standard zebrafish diet. We do this in our trout and salmon work. The details of the feed should be provided. Like I said, formulation, source of ingredients, their composition. And again, I'm going to say this Miller paper really impressed me. It's a good paper. And then the nutritional requirements for three life stages should be identified. That kind of worried me a little bit. These zebrafish grow so fast. And they're in other species of fish, there are really three distinct life history stages. You got to put it like this: baby fish, juveniles, most juveniles, and then reproductively active fish. So that's something that needs to be investigated. One good bit of information or good development is that there are related cyprinid species that are being grown in great numbers now in China just over the last 10 years. They're grown for food fish, they're bigger fish, but they're related. And so maybe this could be a starting point for looking at some of the nutritional requirements for zebrafish. Oh, rats. Can I go back one? So the positives, you do have some good semi-purified feeds. Again, just to repeat, I think it would be very easy, not easy, but it wouldn't take very long, I don't think, to at least identify and quantify the nutritional requirements of zebrafish. Interesting thing about zebrafish, you can do studies pretty fast. Short life history, you can do 15-day feeding trials and probably work some of these out pretty quickly. Another advantage we have, a positive, is that we have new response variables compared to the early salmon days when it was basically weight gain and survival and other easily to measure metrics. And the other thing that I thought about was we kind of know the boundaries now of some of these nutritional requirements. So we can narrow in more specifically to the levels that we think are important without starting from scratch. And again, as I said, others I printed are root for food, fish, and that will get us off to a good start. And I really like this workshop. I'm excited to come here. I've been thinking about this for a long time. at least 10 or 15 years saying, what's with those zebrafish guys? They really, how are they doing this work? And I think the time has come that zebrafish nutritionists can really benefit from the accumulated knowledge and experiences that we've developed in the other sector of aquaculture, meaning food fish production. So I think you can jumpstart this and get at it pretty quickly. So with that, I'll conclude and say, just to repeat with my little triangle there that I showed, my pyramid, I think you're now at a similar stage that trout and salmon were in the 1950s, but I don't think you're going to have to wait 50, 60 years to get to where we are today. With the new knowledge and the new connections we have, with a few years of effort, we can catch up and clean things up here pretty fast. So thanks very much.
Thank you very much, Ron. Pulled no punches. Any comments?
I'm old enough I don't have to.
There we go. That's right. Any comments? Yes.
Yeah, I was glad to hear you say that if you got a paper that really did a poor job of describing the fish diet, that you would reject it outright. And I think it's worth saying again, I know a lot of people have already said it, but I think that's one of the problems in general, certainly with the rodent literature, and hopefully we can avoid this with the zebrafish literature, which is papers are being submitted and reviewed by people that don't necessarily have the nutrition background to review them, and so they'll get accepted and they'll compare a cereal base to a purified or something that's, as we're agreeing here, is fairly inappropriate, and they'll get published in a very, top tier journal, and that kind of just feeds forward. And I'm also wondering if this can get backed up to the grant review stage where, folks writing these grants, this stuff can get caught at that stage. This whole rigor and reproducibility thing, we shouldn't really be funding poorly designed studies, especially diet-wise. So that's just, to me, that's just a constant thing you see from a nutritionist perspective in the literature. Bad diet upon bad diet upon bad diet, and it just feeds forward, because then that paper gets cited as the gospel, and now we have a problem because we're citing bad data.
That's easy to fix. At the review stage, if NIH were to reach out a little more broadly into the commercial, or our world basically, we could provide that kind of input. We don't know the details of some of the work that you're doing, but we could certainly provide input on that. The other thing I would say, I would like to see more cross-pollination, less stovepiping between aquaculture nutritionists like Dilbert and Lou and myself and the zebrafish community, because I think both of us could profit from that.
Okay. So just a quick comment about the journal Zebrafish and about your statement too. Steve Ecker, the editor of Zebrafish, has made an effort to bring these kind of topics more into the consciousness of the zebrafish community. And the Zebrafish journal was a vehicle that was best suited for that. But unfortunately, it's not available to everyone because it's not open source, unless the authors can afford the fee. But nevertheless, I think the starting point has been given to make people aware that nutrition is something we have to pay attention to this. And I would be very grateful if the cross-pollination aspect that you mentioned would really happen, and we can, successfully publish zebrafish papers in nutrition journals, and make a contribution to developing better knowledge about this issue.
Well, I just comment that I do see these papers in aquatic toxicology and journals like that. So there is every paper isn't published in the zebrafish journal. There's some that get out.
Just to provide some background, I think one key point is the zebrafish sort of evolved out of an interest in studying early development. And so the thing that was only optimized was, do the fish grow quickly and how many eggs do I get? And so that community started, but then the pressure to address so many aspects of physiology emerged, such that the whole field now is asking questions about cardiovascular disease and heart development and regeneration and a ton of physiology, building on essentially no knowledge beyond this anecdotal, like, okay, you can raise fish this way, that way, or another way. So I think we never had this period of optimizing growth the way a commercial field would have, because you need a adult fish out the end. And the zebrafish for a while didn't care about anything beyond like 48 hours, and even that was late.
That's a good point.
So I think we are, but that said, the whole community is changing because really interesting physiology is there to be discovered with this model. So I think we just have to do somewhere, we have to figure out a funding stream to really do these. Really, we could even start with your tables. Like you said, it shouldn't take us 20 years because we could start with what aquaculture has done and just see how close does the fish match that.
That's a fair point. It has, you're right. It's evolved from its origins to much broader.
And editors, like a lot of editors who developmental biologists, probably know nothing or very little about this concern. Because if you produce an egg and it develops in 24 hours, it's fine.
Done.
Right.
Yeah, good point.
The comment about what you were saying earlier, and let's just make it clear, we don't know what a bad diet is. So we cannot regulate any of that at this point because, as Dr. Hardy said, we just don't know a whole lot about nutrition and what are in these diets and how it affects the zebrafish. So we can't regulate from the front end saying, you know, we're not going to approve your study because you don't have a good diet. But we can say, you know, when you do publish this, publish some nutritional information with it, okay? And that can be as simple as, you know, if we're using a flake diet from the pet store, let's send it in to get analyzed for a quick, even if it's approximate analysis, right? That would be a good start to get the protein, the lipids, and so on and so forth, because we don't even do that at this point. Eventually, we'd like to improve that and get even more rigorous with some of those reportings, but start out very basic until we actually find out what we know is good and bad for zebrafish. So that may be a matter of semantics, but let's try not to, when we talk about this to our fellow colleagues when we leave here, let's not talk about those types of things, you know, because that's not really what we're doing. We don't really know what's good and bad at this point. So
We'll go ahead and move on, and we'll have additional discussion later. Thank you very much, Ron.
Thank you.